Good morning or good afternoon, wherever you are. I wish to welcome you to the second and last day of this Linnean Society International Conference, Evolution on Purpose, Teleonomy in Living Systems. But before we start, just a little about the origins and aspirations of our society. The Linnean Society was founded in 1788, named in honour of Carl Linnaeus. In 1858, the Society famously hosted the first public presentation of the theory of evolution by natural selection, with papers by Charles Darwin and Alfred Wallace. To this day, the Society remains very active in its mission, which is to create a world where nature is better understood, better valued, and better protected. I do hope those of you attending this meeting who are not members will feel inspired to join. Like all societies, new members are vital to fulfill the endless need for reinvigoration and progress. Details can easily be found on the Society's website, which is https://www.linnean.org. So, my name's Dick Wainwright. Welcome again for those just joining to the second and last day of our international online Linnean Society conference, Evolution on Purpose, Teleonomy in Living Systems. We now have another 8 talks, after which I will hand over to lead organizer, Professor Peter Corning, who will moderate the final open discussion section from 17:30 GMT, Greenwich Mean Time, and bring the meeting to a close at or before 19:30 GMT, a couple of hours later. Do remember to keep a note of your questions for speakers ready for the discussion, because there is no, as yesterday, there is no opportunity to raise questions live during the proceedings. So our first talk today is by Professor Armin Moczek from Indiana. The title of Armin's talk, which has been pre-recorded, is When the End Modifies Its Means, the Origins of Novelty and the Evolution of Innovation. So Padma, if you could press the button. We haven't got any sound, Padma.
During the actual
Sorry,
event,
1 second, I'm just going to restart it.
Memorial service. All right, thank you all for coming. I want to start by thanking the organizers for giving me the opportunity to give this presentation and to give it in a recorded fashion. I'm actually, as you're listening to this during the actual event, participating in a memorial service. So I really appreciate the opportunity to pre-record this presentation. All right. When I was a young graduate student, one of the questions that consumed me concerned the origin of novel complex traits. I was interested in the first eye, the first wing, the first light-producing organ, the first placenta, traits that once invented, revolutionized life on this planet. I wanted to know how these traits come into being. What are the origins, the baby steps of innovation that launch these major revolutions? And the classes I took back then introduced me to 3 frameworks meant to emphasize what matters most in evolutionary biology. So, for instance, I was introduced to the framework of evolution by natural selection, which views organisms essentially as Swiss Army knives, as an accumulation of gadgets, each honed to execute specific adaptive functions. I was also introduced to a historic framework, which emphasized that yes, adaptation is important, matters definitely, but what matters even more, perhaps most of all, are random events, historical accidents that wipe out genotypes, populations, entire phyla, regardless of how adapted they were or not. And lastly, I was introduced to the framework of developmental constraints, which would emphasize that the overarching force in shaping why evolution truly unfolds the way it does is essentially one of engineering, determining the phenotype space accessible or not by what development allows or not. Pigs will never fly, no matter how adaptive that may be or fun. Okay, now these frameworks, of course, are not wrong. They remain highly important and influential. They're, of course, non-exclusive of each other. But very quickly, I also realized that they are equally useless when it comes to understanding and answering the question I was after, namely the origins of novel complex traits. Why is that? Well, natural selection cannot select for something that isn't there yet in some way, shape, or form. And historical accidents can only sort among pre-existing variation, but by themselves, they do not create new opportunities. And the same is true of developmental constraints, which can only veto options, but not create new ones. And so as a consequence, these frameworks, no matter how helpful in other contexts, have not really helped us advance our understanding of how novel complex traits originate from within the confines of ancestral variation. So the origins of the first eye, the first wing, the turtle's shell, the first bioluminescent organ, the mammalian placenta remained unapproachable by these existing frameworks. And in fact, I would say that by prioritizing these frameworks, conventional evolutionary biology has now essentially moved away from even asking that question. Not because it's not an important question. I would actually say it's one of the most important questions in evolutionary biology, but because conventional evolutionary biology lacks the tools for addressing it. So it became clear to me that in order to make headway, novel ways of thinking and methodological approaches were needed. And the rest of the talk, I'll focus on the contributions of evolutionary developmental biology and how it has helped our understanding of the origins of novel complex traits and the process of innovation in evolution. And specifically, my thesis for today is as follows. It is that evolutionary developmental biology broadly, but especially the concepts of developmental bias, modularity, reciprocal causation, and constructive development provide highly promising opportunities to revisit how we conceptualize and therefore study the origins of novelty and the nature of innovation in evolution. Okay, so this is where I'm headed. To develop this thesis, I will first revisit several key contributions Evo has made to our understanding of the origins of novelty as I see them. I'll then switch gears and try to convince you that the means by which development facilitates innovation is itself a process that is evolving and subject to continuous innovation. And I'll end by briefly touching on current challenges to our understanding of the genesis of novelty as I see it and the nature of innovation that we have to overcome to make further progress. All right, so for the first part, I would like to focus on two topic areas in particular. First, I'd like to emphasize the conserved as well as the modular nature of development and its implications for the process of innovation. And then I would like to say a few words about the reciprocally causal relationship between phenotype construction, robustness, and plasticity. All right. Okay, so arguably one of the most fundamental contributions of Evo has been the realization that the extraordinary and awesome phenotypic diversity that surrounds us on the level of external and internal morphology is not underpinned by a corresponding diversity in genes, pathways, morphogenic processes, etc. Instead, the developmental basis of phenotypic diversity is remarkably conserved. Divergent organisms rely on the same developmental means, the same genes, pathways, morphogenic movements, etc., to instruct the building of both similar and very different traits. This realization has forced us to reconceptualize organisms less as completely unique products of ontogeny, like these four taxa depicted on the right, and more like unique assemblies of the exact same shared and deeply conserved building blocks, able to be reassembled to build both similar and different traits. Okay, let's quickly look at some specific examples to drive this point home. The eyes of vertebrates arthropods and cephalopods are convergently evolved organs, yet their development relies on the same set of homologous transcription factors, opsin proteins, cell types, and neural circuits. The pump that's pumping in your chest at the moment and the dorsal vessel of insects have nothing in common. They're independently evolved contractile organs, except their development, of course, relies on the same homologous transcription factors and cell types. And I shouldn't say they have nothing in common. They seemingly have nothing in common unless we look at how they're built. And perhaps most extreme and dear to my heart, projections, sticky outies, as diverse as the siphons of ascidians, the tail of a mouse, the tube feet of echinoderms, or the leg of a cockroach, again, superficially seem to have absolutely nothing in common. I mean, they're all belonging to very different phyla, except once we look at the organization of the proximal distal axis, we realize it's organized by the exact same set of deeply homologous transcription factors. Initially and conventionally, and partly to this day, findings such as these are interpreted as evidence of developmental constraint, right? The notion that development only allows for one way to make an eye, a heart, a limb, thereby, of course, limiting evolution. Ivo Divo has expanded this perspective towards the notion that rather than just constraining evolution, the nature of developmental processes provides a set of pre-existing opportunities to evolve complex functions repeatedly, reliably, and regardless of exact taxonomic context. Okay, this of course then begs the question, what is it that allows developmental processes to be so extraordinarily reusable? Okay, it is at this point that I would like to switch and introduce you and emphasize the modular nature of development and its implications for the process of innovation. And I could fill a whole day just talking about that, and sometimes I do, but this time a few minutes will have to do. So I'm going to introduce you to what I call the 4 H's. The modular nature of development manifests, of course, as all of you know, in heterotopy, or an evolutionary change in developmental location, thereby allowing old developmental modules to function in a new domain. For instance, recruiting the module that, as shown on the left, ancestrally determined the proximal distal axis of conventional appendages, such as legs, to the pronotum of beetles, facilitated the evolution of beetle horns. The modular nature of development also manifests in heterochrony, an evolutionary change in developmental timing, thereby allowing old developmental modules to function at a new or different time relative to other such modules. This is obvious, for instance, in the evolutionary change in the timing of limb growth, which allows bats to develop relatively long and flightless birds to develop and evolve relatively small forewings. Now, that seems like a simple or relatively moderate evolutionary change, but heterochrony can also bring about rather profound transitions, such as in the evolution of holometabolous metamorphosis shown at the bottom, from incomplete hemimetabolous metamorphosis in insects shown at the top, for the relative expansion and contractions of specific life stages, resulting in one of the most impactful radiations in the animal kingdom. The modular nature of development further manifests in heterometry, an evolutionary change in the amount of developmental product. And even though this seems like a simple change, if it involves growth factors that specify beak shape, changes as simple as these by themselves, in this case, 2 particular growth factors, are alone sufficient to generate the full range of beak sizes and shapes in Darwin's finches. Lastly, and I do realize I'm probably only one of 2 people on this planet who uses this term, there is heterocyberny, or an evolutionary change in the governance of a process, the other person being Scott Gilbert, who coined the term, or put another way, an evolutionary change in the upstream regulation of an otherwise downstream and very conserved developmental process. I think this term is important, or at least the concept is. Heterocyberny is ubiquitous. The more we look, the more we see it. And it can, of course, again, be very profound. Just one example very quickly. Think of somatic sex determination, which is ultra conserved across phyla, but the upstream mechanisms controlling it are all over the place, meaning they're extraordinarily evolutionarily labile. Okay, the conclusions I want to draw from this first part are the following. The combinatorial reuse of modular conserved elements across contexts facilitate the evolution of diverse and novel regulatory settings and phenotypes without having to evolve novel regulatory machineries or building blocks. Okay. Now, this deeply conserved modular and combinatorial nature of development also has important implications for our understanding of the relationship between phenotype construction, robustness in development, and developmental plasticity. Why is that? Well, for starters, we have learned over the past couple of decades that phenotypes construct themselves in a sense that every round of phenotype construction creates the developmental environment for the next round of phenotype construction, or if you flip it around, each round of phenotype construction is responsive to the kind of phenotype construction that took place in the preceding round. As a consequence, all of development, one way or another, is responsive to context. When I was a graduate student, I learned that there is development and then there is context responsive or environment sensitive development. That is nonsense. All of development, to develop, means to be responsive to conditions, internal, external, or both. Now, when this context responsiveness is combined with modularity, it has great capacity to facilitate trait integration and functionality. For example, during vertebrate development, muscle precursors migrate randomly, but are maintained only in positions relative to concurrently forming bones. Muscle precursor cells are responsive to the presence of bones. This is where they change their development and settle and form muscular skeletal connections. Same is true for motor neurons, which proliferate abundantly during early developmental stages, but they are maintained once they sense the presence of muscles to be innervated. Same goes for the vascular system. It expands into empty space during early embryogenesis, but where it senses hypoxic conditions, it establishes itself further. In each instance, complex and discrete developmental processes integrate with each other through reciprocal interactions, thereby creating, forming higher order levels of organization, but this is achieved without the need of a higher order organizer. Rather, integration emerges through the context responsive action of modular component parts. Lastly, this context responsiveness, coupled with modularity resulting in integration, then allows developmental processes to respond to perturbations in a manner that yields adjusted, but nevertheless integrated and functional phenotypes. In the process, living systems are afforded the ability to adjust to stressful environmental conditions, including conditions perhaps never encountered before in their evolutionary history, without necessarily falling apart. Instead, they adjust, and sometimes these adjustments may bring about significant innovation. Okay, time for a quick example. There's a growing number of these examples. This just happens to be one of my favorite ones. Polypterous fish can be used as a proxy for vertebrates prior to the water to land transition. When these fish are reared in a terrestrialized environment, meaning they have to walk on their front limbs rather than swim, within a single lifetime, they adjust and improve not just their behavior, gait, and posture, but also their skeletal features. And these adjustments parallel the fossil record of tetrapods, ascendants onto land. Okay, there's a growing number of such examples, and collectively, they suggest that two things: interactions between developmental systems and environmental circumstance may bias the production of phenotypic variation in the face of novel and stressful environments, and not just bias it in any particular direction, but bias it in the direction of functional, integrated, and possibly adaptive variants. And if that is correct, it suggests that novel phenotype production in development through interactions between developmental systems and environmental ecological circumstances may well precede rather than follow changes in genotype. Okay, So with all this in mind, I now would like to move on to the second part of my talk. So far, I've tried to emphasize that what you might call the nature of development scaffolds the way evolution innovates. Now I would like to try to make two additional points. The first one being that the scaffolding itself evolves over time. This nature of development did not all come into being all at once. Instead, it was added over time to the developmental repertoire of living systems. You can pick your own favorite starting point of this sequence. Mine is when cells decided not to separate after dividing and instead to form multicellular epithelia, which then acquired the ability to allow some members, some cells to migrate, creating 2 and eventually 3-dimensional shapes in the process, adhere to substrates and to each other, maybe respond to the fusible morphogens to organize themselves into more distinct regions, aided eventually by Hox genes and their products to form segments, but also the invention of specific cell types and the organs they ultimately were able to generate, such as the neural crest, or organs such as the amnion or the placenta, which fundamentally change what organisms can do, including how they interact with the environment, each other, and their offspring. Everything listed here, of course, operates on different levels of biological organization. I get that. But what they all share is that even though they're all products of evolution, once in existence, each one of them fed back to influence subsequent evolution by adding unique degrees of freedom with respect to what development could do and what kind of variation it was able to produce for evolutionary processes to then act upon. Okay, now comes the second point I'm trying to make. There's no reason to assume that this process has somehow stopped, right? Organisms, developmental systems, including ourselves, continue to acquire new degrees of freedom, for example, through symbioses they engage in, or the ways in which they modify or respond to modify environments. Again, some of these operate on different levels of biological organization, but they all share that they have the potential to bias as well as create novel variations available for evolutionary processes to act upon. All right, so to summarize, this part, I hope to have shown you that the nature of developmental processes not only scaffolds the genesis of novel traits, but that the scaffolding itself evolves over time, thereby increasing the repertoire of developmental systems to pursue a wider range of possibilities and across an expanding range of conditions. And I speculate that, no, I don't speculate. If this is true, then developmental evolution is a process that continuously modifies its own means. What I do speculate is that there's no reason to assume that this process has somehow stopped. Okay, now let me end my presentation by stepping back and highlighting where I think some of the major challenges reside as I see them. So what you're in for is a highly subjective list of sort of pet peeves. Here's perhaps the most important one. I'm part of a discipline, evolutionary developmental biology, which really should call itself comparative developmental genetics. But we have learned that genes and genomes don't make traits. Rather, they supply critical resources for the process. Instead, traits and organisms are the product of developmental systems which interact with internal and external environmental states they themselves help create and maintain themselves. Evolution of development therefore ought to be conceptualized as a heritable change in these interactions. Yet this is not how we study it for the most part. Second, related to this, we need new models and new tools to put to the test and assess the role of developmental bias in shaping selectable phenotypic variation, and to explore the interactions between developmental systems and ecological conditions, to contrast ontogenetic responses to stressful, novel conditions across various scales. Here, I would say we are actually on our way to getting there, and I think there's no better time than the present to meet this objective or this challenge, given on one side the confluence of absolutely awesome novel techniques, molecular bioinformatics, etc., but also, sadly, the ever-growing number of species confronting a rapidly changing planet. If you're interested in studying the interplay between developmental systems and ecological conditions, especially under stress, now you have no shortage of opportunities to do so. Thirdly, I think we need to reevaluate how we conceptualize the environment and its role in development and developmental evolution. Our traditional view emphasizes the environment as something that is external to the organism, separable from it, and passive. You take the organism away, the environment's still there. But over the past couple of decades, we are accumulating increasing insights that make us realize that environments are, of course, shaped by organisms, internal, external environments. Aspects of the environment are inherited in ways that affect developmental evolution of offspring. If all this is combined, exactly where the organism ends and the environment begins, and exactly what the heck is actually evolving, is sometimes a lot harder to say, but biologically a lot more interesting and perhaps more meaningful to investigate. Lastly, do we need a new terminology? I struggle with this. Maybe, or quite likely in some cases, terms such as developmental constraint, genotype, phenotype map, developmental program, I think are, how should I say this, worth revisiting and discontinuing. They're not particularly, I think they're on their, they have become essentially unhelpful. But maybe not. I struggle with the term agency, even though I use it in my own writings, because I'm not entirely sure what it adds that is not already captured by niche construction, especially if niche construction is allowed to happen within an organism, and developmental bias. And I'm especially skeptical, to be honest, about terms such as teleonomy, purpose, and goal orientation. But this is also why I'm here, and I'm looking forward to seeing everybody else's talk to be convinced of their importance. Okay. And with this, I would like to close and thank my many collaborators who made the work that fueled some of these thoughts possible. I want to thank my funding agency, without whom none of this would have happened, and especially a couple of people without whom I simply would not be here. And lastly, I want to thank you for your attention, and once again, the organizers for giving me the opportunity to give this presentation. Thank you.
Well, I don't know what you thought, but I thought that was a wonderful presentation with many challenges for what this meeting is about. All sorts of very important points there, I think. We are 2 minutes now from past the hour, wherever you are, and we are going to stick to schedule, but there's not a lot of time. Just before we continue, which is with a live talk, I'd like to remind those speakers who are making live presentations that you do have a maximum of 29 minutes. And if necessary, I'll remind you of the time with 4 minutes to go. And again, when you only have 1 minute left. Yesterday, everybody was brilliant about keeping to time, and that is hugely appreciated. Also like to remind people on the presentation panel to try to remember to turn your video off and mute your sound during talks as much as possible. Right. Well, just wait a few more seconds. So I think we're more or less at exactly past, I think so. So our next presentation comes live from New York by Professor Stuart Newman. The title of Stuart's talk is Cellular Agency and Mesoscale Physics in the Evolution of Multicellular Development. Seems like an appropriate title to follow the last. Over to you, Stuart. Thank you.
Okay, thank you. I don't know if my video is...
Stuart, we can't see you.
Yes.
Yeah, you appeared for a second.
Okay.
Yeah.
Got it.
That's good.
Okay. So yes, it was very fortunate that I alphabetically follow Professor Moczek, because I'm going to dive more deeply into some of the issues that he so eloquently raised. So I'll share the screen here. And
Okay, we can see that.
great. Okay, and got it. Okay. So yes, so we know a lot now, and we've heard some of it very recently in this meeting about the history of life, the diversification of organisms, and particularly multicellular organisms. The origin of cellular life is still obscure and controversial and not settled, but we know a lot about these branch points, and we know a lot about the single-celled organisms that were the antecedents or ancestors, a lot of these multicellular forms. And we know about the time course in which they emerged. Some of the diversification happened very rapidly in geological terms. So how did that happen? The information that we have as far as the phylogenomics, as far as the ancestral relations, as far as our knowledge of how the Earth itself has evolved, has allowed us maybe to revisit a question raised by Stephen Jay Gould about whether all this would happen the same way if it were run again. Is there anything inevitable about these changes, about these evolutionary innovations and so on? Or is it simply organisms stumbling on solutions randomly because of the need for adaptation? So we also have definitions of organisms that come to us through kind of history and recent history, organisms as natural purposes, autopoietic systems, autonomous systems. But how do such entities arise during multicellular evolution? Is there anything inevitable about this? And is there anything directional or teleological about the progress that we've seen? Okay. So here's another schematic of the arrangement of the major clades in the history of life. We have bacteria, archaea, and so on. We're down here in the Opisthokonts. We're actually more related to fungi than we are to plants. And here we are, metazoa, a little twig on this tree. And these are the major groups that have multicellular forms. And there may be some elsewhere as well, but they're most prominent in these groups. And we know that the cells that give rise to these multicellular forms are different from each other, and the way the cells attach to each other are different. So the physics of the organizations, the multicellular organizations, are different from each other. So I'm going to be discussing multicellular form, cellular function, and cell agency, and I'm going to come to conclusions that are somewhat at odds, not so much with talks at this meeting, but with the standard narrative as it developed through the 20th century. I'm going to suggest that multicellular forms are in some sense inherent to the materials that build multicellular structures, so that there's an inevitability to their the playing out of their morphological diversity. Cell functions, we know less about. How do individual cells, ancestral cells, how do they gain the functions that they did? But multicellular organisms have functions that are related to those ancestral cellular functions. And I'm going to talk somewhat, because there's some new information about this, about how those ancestral cell functions have been appropriated by multicellular organisms in their evolution and development. Then I'm going to be talking about cell agency. We know from this meeting and elsewhere that individual cells themselves have agent-like properties. They explore their environments. They're not simply passive. Okay. So my proposition is that much of multicellular evolution, rather than arising from cycles of gradual selection for adaptive advantage, was driven by the inherent physical properties of multicellular matter, the optional recruitment of ancestral unicellular functions to specialized cell and tissue types, and the progressive alignment of ancestral single cell agency to the emerging motives of multicellular entities. So there's a lot of language here. It'll become clearer as I go on. First, I'm going to talk about the inherency of multicellular form. So we can look at different life forms, and we can look at things like slime molds, and these are cells that are embedded in a kind of a amorphous material. They're kind of sticky and flowing materials. They're not quite liquids, as I'll point out, but they can give rise to somewhat elaborate multicellular forms. I'll jump over to here, and we have plants. And plants are cells that are embedded in solids, in solid matrices, but the cells are very active. And we heard earlier in this symposium that these organisms have agency, and their cells have agency, but they're not moving. They're communicating with each other through what are called plasmodesmata. This is a very active material, but it's nothing like other materials we understand in the non-living world. We don't have a good physics of these materials. If we look over at our ancestors, the non-metazoan holozoans, there are multicellular forms here, but they are stuck in matrices, and they're more or less solid. The cells are not really changing their position very readily. But the metazoans, our clade, are liquid-like. The cells are directly attached to each other like the subunits of liquids, but they move readily past each other. And we know a lot about the physics of liquids, and liquids have very interesting properties if their subunits are polar, if their subunits are elongated in shape, and so on. They can form liquid crystals. So we will look here at the non-fungal opisthecons, the choanoflagellates, the ichthyosporans, and the metazoans, and note that one or more of the extant holozoans, and by inference, the unicellular ancestors of the metazoans, contain genes specifying a whole range of components that are part of the metazoan developmental toolkit. But those don't act in multicellular development because these are primarily single-celled organisms. But when these organisms came to interact, when these cells came to interact with each other in the metazoans, they formed a new kind of material. And that material was a liquid-like material that allowed cells to cluster. Well, these are the ancestral clusters. But in the metazoans, some new genes emerged, classical cadherins, which mediate connections between the cell surface and the cytoskeleton. So these cells remain coherent while they move past each other. And you get the possibility of new kinds of structures as soon as this happened. They're latent. They're not all evident immediately. And then when you acquire some new genes, genes that allow the basal lamina to be made, an enzyme that polymerizes collagen, or genes that allow the cells to acquire a polar shape, you can get many more complex structures essentially for free, because the interactions within the materials have morphological capabilities that are not evident immediately, but with changes in conditions in the environment and external physical conditions, these become manifested. So we're familiar with the idea that non-living materials exhibit characteristic morphological motifs that depend on their composition and the mesoscale or middle scale physical processes mobilized by the respective materials. So liquids can form waves and vortices, and minerals can form characteristic structures. And these are very reliable, reproducible structures that are characteristic of the material. Well, the biological materials, particularly in the liquid-like metazoan or animal tissues, have a number of attributes. We call them dynamical patterning modules. They're specific molecules and pathways in association with specific physical forces and effects that pattern and shape multicellular aggregates. So most of us are familiar with the idea of gene regulatory networks. Gene regulatory networks are very suitable for changing the phenotypes of individual cells. So you can get different cell types by the action of gene regulatory networks. But dynamical patterning modules not only include gene regulatory networks, but they include physical effects that allow the molding of these masses into three-dimensional structures. So you need the physics in addition to the gene regulation in order to get those forms. And the forms that you get are reliably produced, and they are repetitively produced in the sense that different lineages of animals have segments and have hollow structures, have multi-layers and so on. So these are expected. They're directionally produced. The most fundamental of these dynamical patterning modules is aggregation, where the cadherins cause the cells to cluster. And the inverse is epithelial mesenchymal transformation, where the clusters fall apart, which happens at certain stages in embryogenesis. So by mobilizing mesoscale physical effects, ancient molecules took on new functions in the multicellular context. So here's a chart that schematizes the different motifs. We call this a pattern language, following the architectural philosopher Christopher Alexander. The dynamical patterning modules have acronyms, but they also have molecules that mediate them. But it's not only the molecules, it's the physics that the molecules mobilize. And it's the physics of the middle scale. And you get the possibility of alternative cell states. When you have lateral inhibition, you get phase separation, multi-layering, topological changes, and so on, segmentation, pattern formation by gradients. These are all physics that are being mobilized by gene products characteristic of these materials. So we can see this actually operative. These are two embryonic tissues, one pigmented, one not, and they form reproducible multi-layers when they are confronted with each other. And it's very similar to what happens when you confront droplets of immiscible liquids with each other. They form interfaces with characteristic shapes and engulfment relationships. And Steinberg and his co-workers, and then following up by more recent investigators, looked at sorting out and fragment fusion as basically driven by the thermodynamics, driven by the physics of liquid-liquid interactions. And we find that these tensile forces of the liquid tissues actually govern germ layer organization. So you can show experimentally that this is actually going on in embryos. In some embryos, these physical effects are superseded by other effects, and I'll talk about why that happens. But there's basically a drive to organize in this fashion. Similarly, if cells are polarized so that they're less sticky in certain portions than others, they will spontaneously organize into structures with lumens or interior cavities. And a similar thing happens in non-living liquids. You get micelles with spaces inside. And you can see that experimentally, the self-organizing properties of mouse pluripotentists initiate morphogenesis, and you get these internal cavities simply by this sorting out of cells that are polarized in their adhesive properties. So if we look at these dynamical patterning modules, they actually were acquired because the corresponding genes were acquired sequentially, and you get more and more complex organisms over time as you acquire additional dynamical patterning modules. Extracellular matrices are solid materials that interact with the liquid materials of the tissue and cause them to undergo certain mechanical transformations. So you get more and more elaborate structures. And finally, you generate this range of different types of animals as the different features, symmetries and coelomes and pseudocoelomes and segments, which happened a couple of times, and other symmetries came in. So this is all basically inherent in the liquid tissues by physical organizational effects. But it doesn't stop there. We know that genetic evolution continues. So even though, and this is very schematic, it's not meant to represent any real organisms, but you can have plasticity of form, which is determined by a set of toolkit genes and environmental effects and inherent physical effects acting in relation to those environmental effects. And you can get transformation among morphotypes by plasticity. But as evolution occurs, you don't necessarily get changes in the forms, but you get what's called developmental system drift. You get substitution of genes for other genes. You even get new physical effects consolidating these forms. And finally, in contemporary organisms, you may have forms that are just have dedicated genetic circuitry that is involved in producing them. And there's no hint of the transformations that you might have seen earlier on because of plasticity. So genetic evolution continues, but forms can be exhausted because a certain type of material doesn't have unlimited forms. Okay, now I'm going to be talking about the appropriation of cell function. Usually function and form are thought of hand in hand as adaptations and the results of adaptive evolution. There are different kinds of ways of looking at functions. Functions as selected effects propagated by reproduction. Traits exist as necessary consequences of an organism's adaptive history. You have functions as causal roles. Organisms are thought of somewhat as machines, and traits exist as requirements for the organism's survival and coherence. I'm going to be advocating a view functions as appropriations of life-sustaining single-cell activities facilitated by metazoan-specific, animal-specific mode of gene regulation. It's inherent to cellular life, these functions, but they are optional for the cell types of multicellular organisms. So just very briefly, the nuclear composition and organization in metazoans is very different from anything that we see in any other kind of organism. Even the immediate antecedents, the non-metazoan holozoans, don't have anything like this. This is a kind of a glassy-like material made up of domains of unstructured proteins that are glass-like or liquid-like, and genes kind of come in and out of these domains, and they are active or inactive in relation to their occupation of these domains. Enhancer sequences that are very distant to the genes that they're regulating congregate in these domains. And this is an undifferentiated cell, a differentiated cell. These domains are called topologically associated domains, and different enhancers and genes that specify proteins come in and out of association of these domains. This is completely unique to animal gene regulation. And I discussed this in this paper, and I don't have too much time to go through it, and actually not that much is known about it. But one point that I try to make here is that the ancestral cells have functions. The functions are already coordinated with each other, and there are transcription factors called pioneer transcription factors that open up chromatin, and they're unique to metazoans most typically. And then there are lineage determining transcription factors that act in coordination with the pioneer transcription factors that give you different cell types. But the interesting thing is that some of these, many of these functions pre-exist in single-celled organisms, presumably the ancestral organisms of the metazoans, and they're just appropriated by this new genetic regulatory system so that they're emphasized or amplified in cell types of single-celled organisms, of multicellular organisms. So in some sense, function is not something that's acquired by adaptive evolution, but function is appropriated in the formation of multicellular organisms, and then they have new capabilities to do new things, but they haven't acquired them by kind of incremental steps of adaptation. The one that's understood best is this very fundamental multicellular function, which is aggregation. And there is a gene, a protein called grainy head, and it's involved, there are homologs in ancestral forms. And grainy head is both a pioneer and a lineage determining transcription factor. And when it's activated, you have different kinds of epithelial morphologies in multicellular organisms. And when it is inhibited, then the cells fall apart and you get epithelial mesenchymal transformation. And grainy head is almost unique in that it's both a pioneer and a lineage determining transcription factor, but there are ancestral cell functions like cytoskeletal mediated motility, electrical excitability, neurosecretion, detoxification, things that relate to animal cell types that if you look at these references, you see that they already exist as coordinated pathways in ancient cells, or presumably ancient cells, at least the modern versions of those non-metazoan holozoans. So basically, these functions are preloaded in the ancestral cells.
Stuart,
So,
you've got 4 minutes left, I'm afraid.
I'll do it. So to
Thank you.
recap, or origination and evolution of form based on inherent morphological motifs of multicellular matter. The genetic realization of morphology is transformed over time by gene regulatory network rewiring and developmental system drift. Forms are inherent to multicellular matter, not solutions to fitness challenges. Origination of cell types and tissue functions based on inherent life-sustaining functions of ancestral holozoan cells, generation of novel cell types by the metazoan-specific gene expression hub system appropriates these functional traits. They're optional. They also are not solutions to fitness challenges. Okay, the last point, the alignment of cellular agency. Let me see if I can. Okay, this is not running, but this paper, if you check it out, you'll see that cells do unprecedented things that they didn't evolve to do. They can go through mazes, they can look around corners and so on. So cells, if all of these multicellular lineages have agency, and colleagues and I looked at these two organisms. This is a prokaryote, which is Myxobacteria xanthus, and this is Dictyostelium discoideum, and they have similar life cycles, even though this is a prokaryote and this is a eukaryote. And the parallel evolution of these morphologies involves cell agency, the ability of cells to kind of sense their environment, and it's scaffolded by mesoscale physical effects. And we wrote about it here, so the interplay of mesoscale physics and agent-like behaviors. So the determinants of metazoan evolution body plans derive from inherent physical properties of multicellular materials. Cell and tissue functions are based on pre-existing life-sustaining activities, and the agency of ancestral cells contributed to the evolution of development. The origin of this cell agency is poorly understood, but is the foundation, we believe, of multicellular agency. And in light of the inherencies of form, function, and agency, evolutionary change was both innovative and predictable. The role of natural selection was limited, restricted to culling of organismal novelties. Evolved organisms resist selection by seeking affordances and inventing niches and new ways of life suitable to these attributes. And I'll just end with a new initiative that's recently been informed will be supported. My colleagues here that have contributed to this work will be involved, and the Evo Devo community will be invited to be part of the program. The details will soon be announced. Thank you.
Well, thank you very much, Stuart. Fantastic. I feel I need another lifetime. some real embryology there, and some really important questions and points in relation to our overall program. So thank you very much for that indeed.